So your choices are: it occurs only in prokaryotes, it doesn't require DNA replication, it produces two genetically identical cells, and then D is the chromosome is circular. So everybody's saying C, that is correct. I know I repeated that a few times. It's going to give you exactly identical, two genetically identical cells. Yes, Kawa, it's C. He has a question mark after C. But yeah, it's going to give you exact same genetic information for the daughter cells as the parent cell. Now we're going to move on to eukaryotic cells. Eukaryotic cells are a lot bigger. They go through mitosis. They do not divide by binary fission. They divide by mitosis. So we're going to cover mitosis next. On here, you can see that the DNA is kind of in the middle here, and it's kind of loose in the nucleus. If you know the enzymes had to read the information, they won't really know where to start and which way they need to go. But here you can see that DNA has condensed into chromosomes. So here the information is organized. If the enzymes had to come in, they would be able to read the information. So there are different phases where this happens. So we're going to talk about that next. So again, to repeat the summary of this slide, eukaryotic cells do not go through binary fission. They go through mitosis, and the DNA is usually, so if the DNA is like this, it means it can't be read. In order for the DNA to be read, the information needs to be organized into chromosomes. So DNA replication and protein synthesis occur when DNA is loosely packed. DNA replication means it copies itself and then it arranges itself in chromosomes. So cell division, mitosis happens when DNA is condensed into chromosomes. So going back, DNA replication happens when DNA is loosely packed. Mitosis means when the cell divides and becomes two cells, it's going to happen when the DNA is in chromosomes. The DNA wraps around something called nucleosomes. So groups of histone proteins. So these are histone proteins. The DNA wraps around that, and that's how it's organized. So that's how it becomes condensed. It starts to organize here. You can see these balls, they are histones, and the DNA is wrapping around it, and it's getting more and more condensed, and that's called chromatin. So eventually it's going to be very, very condensed. So before cell division, nucleosomes cluster together into chromatin. Chromatin means it's going to start getting packed like this, and it's going to turn into a chromatin. So chromatin means it's tightly packed. So it's kept tightly packed for cell division. And then here, if you keep following, these are the histones, and the DNA is wrapping around it, turning into chromatin. And this part of the chromosome is called chromatin. This is chromatin. This is another chromatin. That's another chromatin. This is another chromatin. And this X letter looking like structure is called chromosome, and which is replicated. And there's one more word I need you guys to know. That's centromere. This part in the middle where they're attached is centromere. This DNA is highly packed, it's organized, it's condensed, so it takes up less space, and it's easier to move than unwound and loose DNA because it's densely packed. Now it can be easily moved around. And I'm covering this because we're about to go through the cell division. So that DNA replication is happening in the interphase. So cell cycle describes events that occur in round of cell division. This is a cell cycle. So we're going to cover the cell cycle. On this picture, first thing I want you to notice, this orange part is interphase. 90% of the time, cell is in this phase. When it's about to either die, then it starts to divide. The cell division is called mitosis. It's only, very small part of the cell life cycle. So this is cell division, which has different phases. We're gonna go over that. And then there's this word called cytokinesis. Cyto means cell, kinesis means to move. So I have another slide on this. So that's when the cell divides. In the interphase, you have three phases, G1, S, and G2. G1 phase stands for growth phase. So when the cell divides here, so we haven't gone over mitosis yet. So let's say the cell divided here. Now you have two daughter cells. Those two daughter cells are going to be smaller than the parent cell. So they need to kind of start growing. So if I were to draw that, so I'm gonna say this is your parent cell. And once it goes through cell division, it's gonna give you two daughter cells that are a lot smaller in size, and these cells need to grow. And they go through G1 phase. So this, I'm gonna say half of the size of the parent cell. So these are daughter cells. And this is the parent cell. So this is just to kind of give you a visual. So when the cell divides here, you have two daughter cells. They're going to be small in size. Therefore, they go through G1 phase. And that's where the cell is basically functioning normally, doing what it needs to do and growing. Then it goes through S phase. S phase is when the DNA replication happens. So S phase is DNA replication, and then there is G2 phase. G2 phase is there's some additional growth that needs to happen before it divides. So that's also growth phase. So G1 is growth phase, G2 is growth phase, S phase is where replication happens, and S stands for synthesis. Let me type that for you. So S means synthesis. That's where the DNA replicates, DNA replication or cloning, if you know it like that, happens. And then the word cyto, I also want to type that for you. Cyto means cell. And then kinesis means movement. So what's happening here? Cytokinesis, cells going in opposite direction and splitting into two. So cytokinesis is cells growing in opposite directions and splitting into two. So during interphase, the cell replicates itself. That happens in the S phase. And this is when it's going to replicate all the information, and it's going to carry out other functions, and then it's going to grow a little bit more before the cell division happens, which is mitosis. So mitosis is the division of the nucleus. The nucleus is going to divide in half during this phase. Cytokinesis is division of the cell itself. Cytokinesis is not part of mitosis. So it's the cell dividing, splitting into two completely. Mitosis is the nucleus dividing. So the information inside the cell is dividing. So before I do this, I kind of want to write out the names for the mitosis for you.
PMAS.
PMAT, yeah, so for mitosis, there is 4 phases. Mitosis is the cell division part. First one is prophase. Second is metaphase. Next is anaphase. And last one is telophase. So these are the 4 phases that are part of mitosis. This is mitosis. Before mitosis, there is interphase. And after telophase, there is cytokinesis. I want you to keep in mind that cytokinesis and interphase is not part of mitosis. Those are just the steps after and before mitosis. So the way you want to memorize this, you can call it PMAT. So that way you can remember it. Some students call it IPMAT. That helps them remember interphase is part of, the interphase comes before prophase, but then don't confuse interphase being part of the mitosis because it's not part of the mitosis. So PMAT, prophase, metaphase, anaphase, and telophase. So let me show you the images for this now. Okay, so first thing, I want you to notice this blue line here. So here you have prophase, metaphase, and then on top it says mitosis. And this is continuous of that slide and ends here. There's no mitosis for cytokinesis. This is not part of the mitosis. Interphase is not part of the mitosis. So let's look at these images, what's happening on the exam. You can expect 2 to 3 questions on these images. I'll show you the image and I'll ask you the phase. So chromosomes on here, so you're gonna notice there's early prophase and late prophase. So basically why we have this is because sometimes when they look at the cell under the microscope, it's in this phase where it's ready to go to prophase. And sometimes it's coming out of prophase. So they named it early prophase or late prophase, depending on when you look at the cell. So here you have the nucleus. Inside the nucleus is the chromatids. So sister chromatids are the replicated ones. So whole thing is called chromosome. So this nuclear envelope, it breaks. So on this image, you can see it's broken. And these chromosomes are kind of being pulled by something called centrioles. Please do not confuse this centriole with centrosome. So that's together. So that's one centriole and then together centrosome. So think of these centrioles as spider webs. As this nuclear envelope breaks, these fibers, they make protein fibers that hold on to these chromosomes, and they start pushing and pulling until they're in the right place. So I'm going to repeat what I just said. So in prophase, nuclear envelope breaks. Centrioles create these protein fibers that hold on to these X-looking structures and start pushing and pulling until they're in the right place. Then they line up perfectly in the middle. Metaphase. Think of M as middle. They line up in the middle, and these centrioles are still pulling on them, pulling on the chromosomes, and then they break in half because they keep pulling on them. These chromosomes break in half. Now they don't look like letter X, they look like V's. That's called anaphase. Think of it as these chromatids moving away from each other. So they split and they get pulled away from each other. Then you have telophase. During telophase, another nuclear envelope starts to appear around the chromosomes. And then that's the end of the mitosis. And then during cytokinesis, it's going to split in half. So you're gonna see this dip here, cleavage furrow or contractile ring, and it's gonna split in half. And then you have 2 cells. So you have 2 daughter cells. This cell is now gonna go and grow more. You're gonna go to G1 phase, then S phase, G2, and then it's gonna go through mitosis again. Another thing I want you to notice that's being shown here is animal cell and plant cell. So it looks a little bit differently in animal cells versus plant cells. And the main difference that you will see is in telophase here. During telophase, you see this dip in animal cells. In plant cells, you're gonna see this cell plate. So this part is called cell plate. So I'm gonna try to draw this for you now.